Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Randy Blakely will present Rare Misspellings of the Genome, Dopamine Mishandling, and ADHD. The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, we've awarded more than $380 million to fund more than 5,500 grants to more than 4,500 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists who are working to find breakthroughs in disorders such as addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, and suicide. I'm delighted to introduce Dr. Randy Blakely. Dr. Blakely is Executive Director of the Florida Atlantic University's Brain Institute and Professor of Biomedical Science at the Charles E. Schmidt College of Medicine. He's a member of our Scientific Council and was a Foundation Distinguished Investigator Grantee in 1996 and 2005. Today's webinar will begin with Dr. Blakely's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit questions throughout the presentation. I'll present your questions to Dr. Blakely after his presentation, and we'll address as many of them as possible in the time allotted. And now, I'm pleased to introduce Dr. Blakely. Randy, the floor is yours.
Thank you, Jeff. It's really a pleasure to participate in this event. I have benefited over the years as a young scientist, as a mid-career scientist, and now as a senior scientist by the support from the Brain and Behavioral Research Foundation. And it's truly a gift that you provide to our community, to so many people in need. Many of us have family members and acquaintances with mental illness, and we know that research is our best bet to overcome these. So today I will be talking about Attention-Deficit Hyperactivity Disorder, how studies of rare genetic changes in our genome can produce changes in dopamine mishandling, and we believe lead to some of the important facets of this disorder and possibly clues to new treatments. I first want to make sure that my audience is aware that I have no conflicts of interest derived from financial support of the research to be discussed. It's been supported generously by the National Institutes of Health. I have consulted in the past for a number of biotechnology and pharmaceutical companies, as noted on the slide, but they're not connected with any of the research I'll talk about today. Now, the work I'll talk about is truly a team effort. The members of the laboratory that I note under the Blakely Lab tab there have all contributed in one way or another to our studies of dopamine in relation to ADHD. And I've been delighted to have outstanding collaborators throughout this work who've made significant contributions. And I just want to highlight a few of the people from my group who have provided the data that will appear today as I talk about our research. Now, by way of outline, I'll cover three major areas. We'll talk a bit about ADHD and its diagnosis, its genetics, and why we think dopamine and changes in dopamine in the brain may be involved. I'd like to then move to our own efforts to identify genetic changes that can alter how our brain handles dopamine and how it acts in the brain. And then our efforts to move from a hypothesis to some real data that will show that when those changes are introduced into an animal model, in this case a mouse, that we begin to see some of those features, and it gives us new hope that we may be able to dig deeper into the brain and understand this complex disorder and develop new therapies. So first, let's talk about attention-deficit hyperactivity disorder in its sort of basic facts and statistics. As many of you know, ADHD is the most commonly diagnosed neurobehavioral disorder of childhood. Estimates currently around 8% of the population ages 2 to 17, or more than 5 million in the U.S., have a formal ADHD diagnosis. The diagnosis is achieved solely through behavioral observations, major symptoms clustering in facets of inattention, impulsivity, and hyperactivity. And there's a male bias in the diagnosis of approximately 4 times as many males or boys diagnosed as females. Approximately 60% of individuals with a diagnosis of ADHD receive treatment through medication, and 25% receive no treatment of any form. Lifelong consequences occur if the disorder is left untreated, and these include higher incidence of substance abuse, conduct disorders, low socioeconomic status and employment, and even incarceration. The symptom clusters that provide a diagnosis within the ADHD spectrum cluster around, as I said, inattentive symptoms and hyperactive impulsive symptoms. Many of you looking at these lists will probably recognize facets of your own behavior, even if you yourself have never received a diagnosis. I know in my household, I'm well known for my capacity to lose things and to become easily distracted, perhaps an absent-minded professor. Hyperactive impulsive symptoms such as moving excessively, talking excessively, difficulty waiting turns, and interrupting are also facets of this disorder and lie in the hyperactive impulsive symptom class. At the bottom, you see an attempt I've made to try to illustrate that this truly is a spectrum, with individuals often falling at the middle in what is called a combined form of the disorder, where traits of inattention and hyperactivity are both present. Now, as I mentioned, we all have some of these traits, I suspect, but to achieve a diagnosis, clinical diagnosis, and to receive prescribed therapies, there's more to it than that. One has to have a significant number of those symptoms, and they must persist over time. They need to occur in more than one setting, and they should occur early. Current diagnostic criteria say before the age of 12. And lastly, they must disrupt normal function and quality of life. After all, I can get by pretty well with my distraction and losing things because I have other people around me who make sure that I can find those things and that I get back on track. But for individuals who suffer from ADHD, and particularly its severe forms, this can be very debilitating and can prevent enjoying the full quality of life. Now, let me talk a little bit about ADHD genetics. This is a disorder with significant heritability, as ascertained through studies of twins and through looking at traits as passed down from parents. There are both common and rare genetic variation linked to neural development and synaptic function that have been implicated in the disorder. Often, individuals with ADHD will present with other diagnoses, or others in their family, in their broader family, will have disorders as well, such as bipolar disorder and autism, that we call comorbidities. Shared genetic heritability, meaning that disorders have different diagnostic classifications, actually derive from similar genetic changes, is increasingly being understood to be very common for major psychiatric disorders. And a recent report from the Brainstorm Consortium has verified this as well for ADHD. Now, when we talk about genetic changes, it's important to note that there are several different kinds of changes to our genome that can lead to risk for ADHD. There's common genetic variation that we all possess. We're all very different at the level of our genomes, and we carry very small changes, and most of these small changes have small effects. Some have no effect, but collectively, we believe that this combination of small effects can add up to significant risk for ADHD. Other individuals have rare genetic alterations, sometimes quite large changes in their chromosomes, that can have large effects. An example would be Fragile X mutation, where individuals have significant frequency of ADHD traits. Now, there's also a category of variants, which is sort of a Goldilocks zone for biologists, that involves rare genetic variation of small to moderate effect. This variation can impact risk, but is capable of being modulated by other factors, other genes, and the environment. This variation often can be found in regions of the genome that make proteins. And it's in the construction of these proteins that allows us to get a better grip on how a gene impacts physiology and ultimately behavior. And I'll give you an example of that as we move through. The story now needs to move to a specific molecule in the brain that can be affected by some of these genetic changes, and that is dopamine. Our nervous system is a very complicated piece of tissue, and neurons constitute the signaling components of the brain in which electrical activity is converted into chemical messages. And one neuron talks to another neuron through synapses where signals are passed from one cell to another. One of those messages is the neurotransmitter dopamine. It's a small molecule, no bigger than, say, a molecule of sugar or of aspirin. This is a very small molecule sent from pre to postsynaptic side of the synapse. Signaling by dopamine for many years has been understood to participate in how our brain controls a number of basic behavioral activities and functions. And it is the disruption of many of these in ADHD that has drawn our attention to the role that dopamine may play and dopamine dysfunction may play in the disorder. So, for example, our ability to detect rewards from cues in our environment is under the control of dopamine. Our attention is modified by the levels of dopamine at brain synapses in our cortex. Our ability to learn and to hold information while we execute complex tasks or executive function, also under the control of dopamine. And our activity, our actual coordinated motor activity, is driven in many ways by levels of dopamine. And we can see that in an individual who loses their dopamine in Parkinson's disease and has severe motor effects associated with that disorder. So each of these can be seen to be an underlying normal property, a normal function of our brain that is disrupted in ADHD, leading us to point our fingers at this molecule many, many years ago. Now, I need to talk about a particular protein that contributes to dopamine signaling before we move on. On the left side of the panel, I have cartooned here a nerve ending in purple that is sending dopamine to another neuron here in green. Dopamine is released from this cell and acts on dopamine receptors that are present, these are proteins, on the cell and activates that cell. dopamine can also bind back on receptors on this sending cell, which serves to provide feedback control over release of dopamine. Now, a critical protein in this process is illustrated here in this blue ellipse, and that's the dopamine transporter, or DAT, as I will abbreviate it as we go forward. This protein, this transporter protein, is responsible for clearing out the dopamine after it is released so that the synapse can be, this area is called the synapse, this area can be relatively clean and free of dopamine so that when a signal comes in the next time, the postsynaptic cell can see that as a new packet of dopamine. If there's too much dopamine here, a little bit coming from this side really won't matter. And it's the dopamine transporter acting like a nano vacuum cleaner that keeps these levels very low. Now you can see this in a set of recordings that were published by Giros et al. Actually in 1996, not 2006, looking at mice that either have both normal copies of their dopamine transporter, one copy, or both of copies have been deleted. Now, using an electrode that can measure the level of dopamine and electrically stimulating brain preparation, you can see that there's a very rapid rise of dopamine on that electrode that clears out from the electrode very, very quickly, on the order of a second. If you have one working copy only of the dopamine transporter, remember you have two copies of every gene, what you see is that the height of this dopamine peak is less, and it's beginning to get slower as it's cleared away, here about 10-fold slower. If you lose both copies of the dopamine transporter, this pulse of dopamine now is very slow, indicating that the dopamine transporter can transform this kind of temporal pattern of dopamine after release into this one. So the sharp and signaling that dopamine needs to carry out is best achieved when you have two functional copies of this gene, all working together at all of the dopamine synapses to remove the extracellular dopamine so that new dopamine signals can be sent. This is really what a dopamine transporter looks like, we think. This is a model from work that's been done in the Guo lab in Oregon at the Volum Institute. And it's what we call a crystal structure, a high-resolution structure of this protein. Each of these ribbons constitute chains of amino acids that are moving in and out of the cell membrane, shown here in gold. Outside the cell, or the extracellular world, is the synapse, and on the inside of this membrane is the cell cytoplasm. And you see that in the middle of this protein are nestled a couple of different molecules. In magenta here are molecules of sodium, and in blue is actually a molecule of dopamine. And what happens when dopamine arrives from the sending cell, it lands in a pit made by these helices, like a staves of a barrel, and that dopamine is collected and then pushed through the membrane to clear it away from this side of the cell. So this protein, this transporter protein, is working to remove dopamine through a process that allows for the extracellular space to be clear of dopamine most of the time. Now, why do we think about that molecule and dopamine in relation to ADHD? And specifically, would like to draw your attention to the medications that we use to treat ADHD. These were first implemented because they were found to work, not because we knew much about the dopamine transporter itself. One of these drugs is methylphenidate, or Ritalin, and it is a blocker of the dopamine transporter. And you can see illustrated here that when the dopamine transporters are blocked by Ritalin, no longer is the dopamine able to be cleared, and it builds up at the synapse, causing problems for the next impulse of dopamine to reach the postsynaptic side. Now, another important medication in the treatment of ADHD is Adderall or amphetamine. There are a number of different versions of amphetamines, such as Vyvanse and other longer-lived forms, but this has been the two mainstays in the pharmacological treatment of ADHD. Amphetamine works a little bit differently than methylphenidate. Methylphenidate essentially caps off these proteins and keeps them from removing dopamine. Amphetamine, on the other hand, is a molecule much like dopamine itself that can enter through the transporter and gain access to the inside of the nerve terminal. In the inside of the nerve terminal, it can change physiology of that neuron such that it makes the transporter actually run backwards and send dopamine that has been accumulated inside back out into the synapse. So it's as if you have a vacuum cleaner that is taking up dust and then spitting it right back out into the synapse. So in either case, both of these medications elevate levels of dopamine in the synapse, and therefore it has been theorized that ADHD may be a consequence of insufficient release of dopamine, such that you need to block these transporters to elevate dopamine. That's at least one theory, and we have another, which I'll get to. Now let me say something about the pathways in the brain that use dopamine. Two major ones I wish to draw your attention to. One, in this purple ellipse here, represents neurons in an area of the brain called the substantia nigra. These neurons degenerate in Parkinson's disease, but in the rest of us, these neurons send their projections to an area called the striatum, an area deep beneath the cortex, the outer part of the brain, that's involved in movement control and impulsivity and response inhibition and in learning. Another major area where there are dopamine neurons is a region called the ventral tegmental area, or VTA. The VTA sends its axons, its projections, to two areas that are of importance for us today. One is the nucleus accumbens. This is an area that's responsible for sensing reward and reinforcing signals, so that when things feel good to us, this pathway is activated. This pathway also sends projections up into the frontal cortex, the area of our brain that's involved in the highest levels of cognitive function and that are critically important in attention and in holding information in thought while we execute complex tasks. And both of these pathways secrete dopamine at these terminals and are believed to have be affected in the disorder. Some evidence for this comes about from brain imaging studies. I bring two here for you. These are both from the laboratory of Nora Volkow, and what she has shown in her work is that using probes that can detect in the human brain the presence of a dopamine transporter, you can see that in individuals that have normal levels of dopamine transporter and they're not affected by ADHD, you can see bright red signals here in the areas of the brain, specifically in this particular section in the striatal structures. You can see in an ADHD subject from her work that the levels of the dopamine transporter appear to be much lower. They're not gone as they are in when these pathways degenerate, but they're definitely reduced. Other work from the Volkow lab has gone on to implicate changes in the density of the dopamine transporter in these regions with the ability of the individual to respond naturally to rewards. And I'll come back to that a little bit later. Now, on the genetic side, other evidence has accumulated to implicate dopamine and the dopamine transporter in ADHD. I first want to show you a schematic of the dopamine transporter gene. And here we see on the top line a diagram of a stretch of DNA. And in that stretch, there are these boxes shown here that simply illustrate areas of that gene, the dopamine transporter gene, that make the protein that I showed you winding through the cell membrane. In blue, those are the regions that make protein. We call those the coding regions of the protein. And you can see that constitutes this, when all put together, as they are naturally, we get the full coding region, the full protein of the dopamine transporter. There are other places of this gene that are not involved with making protein, but in regulating how much of the transporter gets made. And so if we look down on this end of the protein, we see an area that's called the 3' untranslated region. And human genetic studies have shown that there are DNA repeats that are variable in number in different individuals. They don't affect the protein, but the number of repeats appears to affect how much of the protein gets made. Now, importantly, changes in that number of repeats have been associated with ADHD now going back since the original studies in 1995 by Ed Cook and colleagues. This work has been replicated in many studies and has led, again, to us to focus on the dopamine transporter and mishandling of dopamine as an important aspect of ADHD risk. So to get to our own research, we took this information from the fact that the drugs that treat ADHD target the dopamine transporter, that genetic variation in the dopamine transporter appears to impact risk, and that dopamine itself works in the brain in areas that relate to the symptoms of ADHD to form this hypothesis that ADHD is a brain disorder that may possess and even be enriched for highly penetrant but overall rare genetic variants that target dopamine signaling, including DAT. And the reason we said rare was because we're very interested in getting to changes that occur in the protein coding regions, which we knew were rare to begin with. We thought that characterization of these genetic variants in vitro and in vivo could allow us to test a hypothesis that changes in DAT and in dopamine signaling might contribute to the traits of ADHD. And thus, having this genetic variation in hand, if we could find it, might allow us to build useful animal models that could elucidate molecular, cellular, and circuit-level mechanisms that actually, at the end, drive ADHD. So in efforts that span the number of years, we went looking for changes in the dopamine transporter gene in individuals with an ADHD diagnosis. And we found multiple examples of rare coding changes, illustrated here, that change the ability of sequence in these areas to encode one amino acid versus another. These amino acids that make up this polypeptide chain are highly conserved between organisms, indicating that they are likely to be very important for the function of the dopamine transporter. And today, I'm going to talk about one of these genetic variants that changes an amino acid called alanine to an amino acid called valine at the 559th amino acid of a long chain that makes up the dopamine transporter. So if you see A559V or VAL 559, I'm talking about a mutation right here in this protein. Now we identified this VAL 559 variant in two boys, brothers, who were young teenagers at the time, with a diagnosis of ADHD. They also had two copies of the high-risk form of the dopamine transporter variants that are in that non-coding region. So we thought that this was particularly interesting and might allow this valine genetic change, even though it's a subtle one for those of you who know your biochemistry, to become penetrant and drive risk. We had a small pedigree in which we found the mutation. You can see the mutation was here, a square means a male and circle means a female. This was inherited from the mother's side of the family and then from the grandmother. We asked the mom and the grandmother to take tests that would give us an indication of whether they might have traits of ADHD themselves. And the mother did not show any obvious enrichment of traits that were amplified in their children, except a mild learning disability as a child. Grandmother, on the other hand, scored above the 90th percentile for impulsivity traits in our measure. And so though this was a small pedigree, we felt that we should pursue this a little bit further and see whether this valine substitution that was occurring was functionally meaningful. And what really drove us to do that was the fact that this mutation had been identified once before, and that was in a girl with bipolar disorder. And you may remember from what I said earlier that bipolar disorder is one of those disorders that occurs in combination with ADHD or traveling in the same families more frequently than it should by random chance. More recently, Jem Sutcliffe and Aurelio Galli's groups have collaborated on a study to show that this variant has also exists in two boys, unrelated boys, with autism. Now, when we first began to look at the dopamine transporter mutant, we expressed it in cells and did what cell biologists and biochemists do to assess function of these proteins. And what we found was that there was normal dopamine transporter protein there. There was normal levels of it on the cell surface. It could transport in dopamine from the outside quite normally. It recognized dopamine with the normal affinity. The affinity for drugs was normal. And thus I had a normal despondent reaction from my graduate student, who, however, had a lot of pluck and perseverance and knew that there were other ways that we could ask about functional status of this protein. And so we hooked up with the lab of Aurelio Galli and found out that, in fact, this transporter is affected by the mutation and in a very unique way. This is an illustration of the setup used in the Galli lab to assess the function of transporters, here shown by these little multicolored circles. Here's a cell expressing a number of these on its cell surface. And dopamine is loaded on the inside of the cell. Rather than putting it on the outside for dopamine to come in, we're loading dopamine on the inside of the cell and watching it come out with, again, an electrode that can sense dopamine. We had no success seeing differences when we looked at how much dopamine came in the cell. But when we looked at whether dopamine came out, which it shouldn't, we found something interesting. And here's some of that data. On the right side here, you see an electrode recording of the signal from dopamine leaving the cell in a wild-type dopamine transporter transfected cell. So this is the level of the signal you see in a normal dopamine transporter. In the mutant dopamine transporter transfected cells, you see that the level of dopamine outside the cell is much higher. And what's more, when we plug the hole, when we plug the transporter with a DAP blocker like methylphenidate or Ritalin, that signal returned to baseline. This suggested that the transporter mutation was allowing dopamine to escape. It was taking it up, but it was then allowing it to escape. When we did studies to mimic the way a neuron behaves when it is electrically activated, which you can do by putting increasing amounts of voltage on the cell membrane, you could see that the difference between the mutant here in red and the wild type in black became magnified. And so when this cell is activated, it actually spits out more dopamine, more and more dopamine. And lastly, if you recall, this ability of dopamine to exit the cell via the transporter is a property of the dopamine transporter when it's been affected by amphetamine. And here what you see is in black that property. You treat these cells with amphetamine and the dopamine signal now increases on the electrode. But in the case of this mutant, remember the signal is already elevated, and I've just brought it down to baseline here so you can see it in combination with the wild type. Now you see that the dopamine level actually decreases. So amphetamine acts very differently on a normal dopamine transporter than it does on the mutant transporter, and in fact, it acts the way a therapy should. It blocks the leak, just like methylphenidate does. This gave us the following model. A normal dopamine transporter here in green is sitting there to clear out dopamine here in orange when it's released from the sending neuron. It comes out and is quickly taken back up, and the levels in this area are very low. In the case of the mutant dopamine transporter, and perhaps in other ways of changing this protein, the dopamine is released, and there are some normal copies where dopamine can be taken back up. But just as soon as it is taken up, the mutant transporters let it back out. And this leak, we think, this dopamine leak and this basal elevation of dopamine is a little bit like not having enough dopamine transporters to begin with. In both cases, you get an elevation of dopamine. It's as if these nerve terminals are being treated with amphetamine all the time. They're spontaneously ejecting dopamine the way they would if you treated them with amphetamine. So that's a theory, and in order to test that, we need to have a preparation with a living brain and in an animal where we can study behavior. And here we turn to the powerful genetics associated with the mouse. Mouse neurobiology has advanced significantly because of our ability to modify the mouse genome and study the consequences. And here we simply illustrate again a dopamine transporter gene. The mouse gene looks very much like our own, and we introduce the mutation where it occurs in the boys, and we end up with a mouse that's carrying just that one base nucleotide that we found in those two boys. So what did we find? When we looked at the levels of dopamine transporter protein in the brain of those mice, you can see that there was no difference, mimicking what we saw when we first expressed the mutant in cells, where there was no difference. When we look at transport activity by putting dopamine on the outside of nerve terminals, you can see that as a function of concentration, the dopamine transport activity increases in the wild type and in animals with one mutation or the heterozygotes, or animals with two mutations or the homozygotes. It's all the same. Their dopamine transport activity is completely normal. But remember, there's something else happening with these dopamine transporters. They're leaking. And even though they can take up dopamine, our prediction is that they would shoot it right back out, and they would elevate the extracellular dopamine. To test that, we took a very small, hair-thin probe that we could stick into the brain and collect a small amount of extracellular fluid in the regions of the brain where dopamine neurons are projecting here in the striatum. And when we analyzed that fluid, what we found was that the wild-type animals keep dopamine levels very low, as expected. But if you had 1 copy or 2 copies of the mutation, the levels of dopamine were significantly elevated, as you can see here. This was proof that, in fact, this mutation could affect the extracellular levels of dopamine and perhaps even drive changes in behavior. Now, I want to remind you a little bit about different pathways of dopamine in the brain, different pathways that use dopamine. For one particular point, first off, I mentioned before that there are projections from the substantia nigra to the dorsal striatum that are involved in impulsivity, in circuits of motor learning, and in habit formation. Then there's other projections that come from the VTA to the reward centers of our brain in the ventral part of the striatum, so it would be down here in this illustration, now moving a little bit further forward in the animal's brain. And they're involved in reward salience. And then lastly, there are projections to the front of our brain, to the prefrontal cortex, that are involved in attention and executive function. I tell you this because some recent findings from our lab show us that the mutation that we've introduced into the animal has specific effects in one of these regions, specifically the dorsal striatum, even though it can be found in all three of these areas. And I'll show you that here. This is a study where we injected dopamine into the brain, and again with a chemically sensitive electrode that could detect dopamine when it was injected, and then when it was removed by the transporters. You can see that the wild-type animals remove the dopamine very quickly, but the mutant animals remove it much slower. There's something in the way that is keeping this dopamine from being cleared. And we believe that is this elevated level of the animal's own dopamine that does not allow the dopamine we injected to get to the transporters, and thus it is slowed down. Now, this occurs in the dorsal striatum, one of those projections. But look in the ventral striatum, the reward center of the brain, and you see that, in fact, there is absolutely no difference here. This tells us that even though mutations produce changes in these proteins throughout the brain, there are certain regions of the brain that are more affected than others. And this is the first clue that we got that truly studying changes in synapses and changes in projections in the animal's living brain would be valuable for us in understanding how a mutation leads to a change in behavior. So let me tell you about a few of the behaviors that we see in these mice. First, with spontaneous and drug-modulated locomotor activation. When the dopamine transporter gene was first identified and then removed from the genome in the laboratory of Mark Carone, some astounding behavior was observed in animals that lost 2 copies of the gene. I don't think I have to tell you which of these animals has lost 2 copies. This animal in black has severe hyperactivity when placed in a novel environment. Here, just moved to a different mouse cage. We could watch this for some time and the animal would still be very hyperactive. Now that excited many of us who are looking for genetic changes that could drive ADHD, but there was a problem, and that occurred later when Manjukurian and co-workers identified humans that had lost 2 copies of their dopamine transporter gene. And what we find in those individuals is not ADHD, but infantile Parkinsonism and dystonia, a severely debilitating movement disorder of childhood. Nonetheless, what we saw here in the mice stimulated our research, much research, into the potential contributions of dopamine and ADHD. And we thought that because our mutation does not cripple the dopamine transport, does not eliminate it, it only causes it to leak a little bit, that perhaps this might be a better model for ADHD, for the study of ADHD biochemistry, neurochemistry, than losing it altogether. In fact, when we took our mice and put them in an open field and measured the distance that they traveled, you see that in relation to a wild-type animal, if you have one mutant copy of ours and two mutant copies, these lines are pretty close together. They're really not very different, indicating that our animals are not spontaneously hyperactive. Now, they are reactionary, and they show that in a motor response we call darting, which is that upon imminent handling of the animals, something that happens to them every time their cage is changed, they show this a rapid move away from the hand, which you can see quantified here, steadily increasing as you get one or two copies of the mutation. And as you see, the peak of this distribution of darting speeds moves to the right the more copies of this mutation you have. And to us indicates that the animals are failing to learn that this threat, this handling, a hand reaching for them, is not going to hurt them. It's going to move them into a new cage and give them new food, is something to be fearful of. What about drug responses? Many of you are aware that drugs like amphetamine or even Ritalin can cause hyperactivity in normal individuals, and yet we treat hyperactive and we treat ADHD with these drugs. So there must be something different about the way these drugs act on normal individuals versus individuals with ADHD. And in fact, what you can see here is that when we give amphetamine to a wild-type animal, in purple you see a normal increase in their motor activation. They run around in the cage. In contrast, if you carry one or two copies of this mutation, you see that the response is significantly blunted. If you inject amphetamine into the animal, either peripherally or directly into the brain, in a wild-type animal, this causes a large increase in dopamine through the mechanisms I've talked about. And you see that in our mutant animals, that does not occur anywhere near the same level. So these mutations, even though subtle, have changed the way not only the animals are behaving without medication, but they're also changing how they respond to these drugs. And we think that might give us clues as to why individuals with the disorder would respond differently to these treatments than normal individuals, unaffected individuals, I should say. Now let me turn to attention, learning, and impulsivity. How do we study this in a mouse? We use a test that's called a 5-choice serial reaction time test, where a mouse is trained to touch with its nose one of 5 lit up panels. You can see them here in a sort of iPad-like device. We train them to touch these panels and specific ones that will be rewarded with a drink of a sweet milk solution back here. We have a signal that will tell them that the test is about to start, and they have to wait for a fixed period of time and then touch the right button. And you can see that here's the mouse. He's gotten the signal. He has to wait, and now he can touch it and go back and get his reward. And then the test is repeated many, many times. In fact, this is a difficult test to run. One has to train these animals for weeks, but ultimately they train up and are quite easily do the task. Now you can see over here in the training phase of their behavior that as they get better and better and better at the task, the homozygotes that carry the mutation are actually training faster than the wild-type animals. And even when they move through the actual test itself, the 5-choice test, we're initially just training them on one of the buttons. But now when they have to do it in amongst 5 choices, they're still learning this task better. Now what happens though when we change the delay, the waiting delay? Remember that impulsivity as a trait of ADHD involves an inability to withhold responses, what we call waiting impulsivity. And here you see that when we change the delay from 5 seconds to 15 seconds, what we find is that the animals show many premature responses. They touch the screen before they should. Instead of touching at 15 seconds, they touch at 5 or 6 seconds. Now, this occurs when the animals have been drilled on a routine regimen of touching the screen after a 5-second delay. And that's the only thing that they ever see. It's 5 seconds, 5 seconds, 5 seconds. Then when you change the rules, they're impulsive. However, if you train these animals with an inability to predict how long to wait, sometimes 5 seconds, sometimes 15, sometimes 10, it becomes a game. You find out that these animals are actually less impulsive. They make fewer premature responses than a wild-type animal. Some of my human psychologist friends have suggested that this may be relevant for how we actually educate our kids who have the disorder, and the literature that indicates that giving these individuals many variable tasks and opportunities in their education and training will actually help them learn better than just giving them routine exercises that otherwise work fine for unaffected individuals. Now, you might say, well, maybe they just can't tell time. Maybe they're having problems telling how long 5 seconds is or 15 seconds. And we actually can test that in what's called a peak interval test. And we assess a distribution of responses after we've trained them on a specific time. And you see here with a 5-second training, they peak in their estimation of time at 5 seconds. When we take this and shift it to 15 seconds, they still estimate, on average, the same time, but their responses are much greater throughout the whole time. It appears that their response level indicate that they are actually more eager to perform the task, to get the reward, than are the normal animals with a normal dopamine transporter. And we've done other studies that indicate that they will work harder for rewards than the animals with a normal dopamine transporter. Thus, we think that there is, as Nora Volkow indicated, something is amiss with their reward system. We don't think it's the actual sensation of reward, because when we test their preference for a sweet solution over over water, we don't see that this difference comes out. It's only in the learning phases of the reward, their willingness to work for it after learning it. It appears to be now much more habitual and unrestrained than a normal animal. Now, lastly, I'd like to just point out that not only have we learned some very interesting consequences of modifying the dopamine transporter, but it has created an animal in which we may find tools that can be used to study humans in ways we didn't think about. Here's one where we realized that we might be able to non-invasively diagnose a dopamine disorder, not by going in the brain at all. After all, that's not something we're likely to be doing with our kids. Rather, we found out that there is quite a lot of dopamine in the retina. And there's a network of neurons, some of which, these amacrine cells in the retina, modulate your sensitivity to light, and they secrete dopamine to do that. And there's a response that when you dark adapt an animal, you place it in the dark for a little while, and then you shine a light on it, these neurons are activated, dopamine is released, and the physiology of the retina changes. You can measure that with a simple surface electrode right on the outside of the eye that doesn't hurt at all. It's done in humans. And when you shine light, you get an electrical impulse that you can record from the retinal activity. And when you quantify that, what you find is that the animals who have 2 copies of the mutant allele, the mutation I've been discussing, can be easily discriminated from animals that carry no or 1 copy. Now, we think with refinement, this measure might allow for a non-invasive inspection of changes in dopamine, certainly in the retina and perhaps in the brain as well. So, what's the future of our work? Is DAT all there is? Well, we know that the dopamine transporter doesn't work by itself. It works together with a number of other molecules, each of which could have its own pattern of genetic variation. And it might be the constellation of risk within this network that drives changes in dopamine in the human brain. But we wouldn't have gotten to this network without having first found mutation and the changes in physiology and behavior that we were able to do by finding the mutation in the dopamine transporter. So let me summarize the findings I've been telling you about. Rare variation in the human dopamine transporter gene has been identified in individuals with ADHD and also in ADHD comorbid disorders, bipolar disorder, and autism spectrum disorder. This mutation exhibits an abnormal outward leak of dopamine that can be suppressed by the two most commonly used ADHD medications, raising the question as to whether or not leak of dopamine may be a central element of the risk for ADHD and could be caused by many different genes. Adolescent male mutant-carrying mice are not spontaneously hyperactive, but they display stress-induced darting behavior and increased struggling when handled, something I didn't mention before, and they suggest deficits in aversive learning or impulse control. These mice increase motivation for reward. They elevate reward-based learning and weighting impulsivity when trained on a fixed routine and where there are predictable outcomes. Although this mutation is expressed throughout the brain, we think that dopamine handling is altered only in a specific area of the brain, a region that's involved in reward learning, habit formation, and goal-directed behavior. Focusing our attention on those areas in individuals who have the disorder as likely to be consequences of altered dopamine handling. And last, these mice may be useful to help us identify biomarkers that can provide for improved diagnosis and a better understanding of the brain in general in the disorder that allow us to generate new treatments that are tailored to those individuals specifically who have dopamine disturbances. And with that, I'll conclude my presentation. It's been a pleasure to talk about our research with you, and I look forward to answering questions as time permits. Thank you.
Well, Randy, thank you for an excellent presentation and for an extraordinary body of work that you and colleagues have done. And I think you really show, in the broadest of senses, the way that science works, from very basic physiology, basic neurochemistry, to genetics, to laboratory animals, and then to how this can relate to human beings and their conditions and new approaches to treatment. So thank you for all that you're doing. One question that people asked relates to the genetics, and at this point in time, are there any genetic tests that a person can have to help with treatment decisions, with diagnosis, etc.?
No, unfortunately, we don't have such tests. The mutations I've discussed today are rare, so you needn't rush out and see if you can get 23andMe or Ancestry.com or your local genetic lab to test for these. There are a few hundred people so far in the world that have been where these mutations have been found. And then more commonly I mentioned that the common mutations that have been seen in the dopamine transporter have very small effects, and collectively you would have to assemble all of those genes together to get us risk, which is a very daunting issue. But so what we think the next steps are is not to try and think about one gene, but to talk about many genes working together. But we think that there must be a convergence of effects of those genes on things like how dopamine neurons develop, how they signal, and how you actually handle and take care of that dopamine. And of course, it's not all about dopamine. I hope I could illustrate for you that some, but not all of the traits that we expect in the diagnosis show up in these animals. There's plenty of room for other perturbations and signaling to contribute to the disorder.
It's interesting that you're able to use this approach to better understand how these medicines that have been used for many years actually work, or have a better sense of how they work. Where do you see this going down the road in terms of even better treatments?
Right. So the current medications and the way drugs are developed is typically one target at a time. It's a very complex and long journey to take a drug from an initial finding all the way to the clinic. And the more specificity you have in that process, the better. And that's oriented us around drug discovery focused on a single target, like the dopamine transporter. And in some cases, that can work well. But as you know, we are not where we want to be in having medications for psychiatric disorders. And so it may very well be that targeting multiple genes within the same network, multiple proteins within the same network, can give us a better, more tailored strategy for drug development to treat these kinds of disorders.
Finally, just I want to ask another question about what I thought was fascinating in the research that you presented, which is the behavior of these mice and changing the time that they have, and that perhaps this fits in with what some people have observed as being when you sort of take a few different approaches in education and tasks for children with ADHD, that may be more effective. I'm curious to hear a little bit more about how behaviors in the mice can relate to behaviors in people.
Right. So scientists are reasonably conservative about trying to transfer our findings from mice to people. We like to talk about it in terms of at least an outcome that gets at the level of behavior, so that we think that these are not just benign mutations. I think all too often we have looked at the behavior of an animal, let's say its hyperactivity, and said, well, that is an indication that the animal has something like ADHD. Maybe, but maybe not. The brain is very complicated, and you can get to the same endpoint through other circuits. Now, I too am quite interested in the change that we observed in those learning studies where the training regimen seemed to influence the outcome just as much as if I gave the animal medication. So I think that this also speaks to other forms of therapy besides pharmacology, and that alone or in combination with medication can help those kids and adults who suffer from the disorder. And this is a discussion that we've been having, and the data has been accumulating for other disorders like depression as well. Medication, along with environmental changes and therapy, make a much bigger impact than our current medications alone. So I think that time will tell as to whether or not the learning responses that we see really do have strong parallels to the human patterns of learning, but they're certainly very provocative.
Well, Randy, I want to again say thank you very, very much for your presentation and for all of your work in this important area. I also want to thank everybody who joined us today. All of the research we fund is made possible through private donations, so please consider making a contribution by visiting our website, bbrfoundation.org, or calling us at 800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, please visit the Events and Webinar page on our website. I hope you'll join us again next month when Dr. Kay Tai, Associate Professor in the Department of Brain and Cognitive Sciences at the Picower Institute for Learning and Memory at the Massachusetts Institute of Technology, will present Lifting the Veil on Neurons that Assign Good or Bad Associations to Our Environment. This webinar will take place on Tuesday, August 14th at 2 p.m. Eastern Time. Once again, thank you for joining us and enjoy the rest of your day. Take care.